And hello, food friends. So, we're going to finish up our discussion of metabolism today and set you up for a little quiz. Hold on a second. Yeah, I gotta go shut the door. All right, so, we're going to set up a quiz, and I'll give you a review sheet and actually summary sheet, open notes, whatever. It should be no big deal. There you go. And I have to say the Flex format has kind of taken me off my stride, and we are way behind. What do you want me to do? Hey, it's COVID. So, we'll finish up in some sort of style, I'm sure. Anyway, if you recall when we last left, we were talking about the breakdown of glucose and how we can use that to generate energy in the form of adenosine triphosphate. And we have really these two primary characters, ATP and ADP cycling back and forth, and NAD cycling back and forth between that and NADH when it has an electron. FADH2 is also there, FAD and FADH2. But the critical part is we have to always recycle things. Now, recycling ATP is nothing. That's what your job is you're always recycling ATP if you're actually carrying out any kind of metabolic reactions as you're growing. But the real critical point becomes, how do we maintain our supply of empty electron acceptors? How do we maintain a supply of oxidized electron acceptors? We got to keep our NAD always ready to go, ready to pick up electrons. So, just to summarize, we had glycolysis. And in glycolysis, we generated a net gain of 2 ATP, and we generated 2 NADHs that are going to have to be recycled. So we make 4 ATP, but it takes 2 to start it. Then, in the Krebs cycle, we make, again, 2, well, it's actually GTP, but we count it in the ATP count. And we're going to require, I think it's something like 5 NADH, and that must be, all right, that's per one cycle. So it must be 10 NADH and 2 FADH2s. So these are what comes out. The last step is the conceptually maybe the most difficult, and I will say that it is in fact the last step of this to be discovered, and it was actually up until the 70s before it was discovered, which you may say still a long time ago, but I actually was taking a class in zoology or something in 1978, 79, and it was still considered a theory. It was not actually understood to be really the way that the mechanism was working. And that's this last step. So rather than listing the last step as sort of a flow like this, it's easier to do as a diagram. This last step, it goes by a couple of terms. It's known as the electron transport chain, which is not a bad term, although it doesn't describe what it really does. It's also known as oxidative phosphorylation. And we'll see here in a bit that's also a really lousy term. Oxygen was required, ATP was being generated. There's a couple of things that I actually have like serious bones to pick with the authorities, and this is one of them. It is referred to as ATP synthesis, and it ain't ATP synthesis. What's happening here is, one, we've got the reoxidation of NADH back to NAD, and FADH2 back to FAD, and 2, we're going to generate what's known as a hydrogen ion or proton gradient, and 3, we're going to use that hydrogen ion proton gradient to rephosphorylate ADP back to ATP. And if you recall, our count was 36 ATP, and so far in the Krebs cycle and glycolysis, we've generated a grand total of 4. So this is going to be doing all the heavy lifting for the generation of ATP. And another way to think about ATP is that really what calories end up being, is how much ATP can you generate from a molecule of food. So in order to really understand what's going on here, we have to have a little bit of cellular anatomy, as it were, and thinking about this membrane-bound structure in the cell called a mitochondria. It's referred to as an organelle. The mitochondria is a pretty big thing inside of a cell. So if we think of a cell like, the size of a soccer ball, then mitochondria are going to be like the size of a large jelly bean. And one cell might have 10, 12, 50, even 100 mitochondria, depending on how much energy it requires. Muscle cells have a fair amount of mitochondria in them. They have 100 or so or more per cell. Now, the mitochondria is a double membraned organelle. It looks like this. So there's the jelly bean, there's the outer membrane, and then there is this real squiggly, highly infolded inner membrane. And inside this space is known as the matrix, and this is known creatively as the intermembrane space. All right. So the Krebs cycle is actually happening in the matrix. Now, what we're going to do is take this spot and blow it up, magnify it up a couple hundred times. So it's going to look like this. We're going to draw that little spot here. There are a bunch of proteins embedded in the inner membrane. So it's going to look like this. We got the outer membrane. We have that big infolding of the inner membrane. And here is the matrix. And this is how it runs. So we've got a series, a complex of proteins that are actually connected by little intermediate things like some of the vitamins. If you're, like vitamin B12, there's a supplement called CoQ10. It's also in here. But these protein complexes are the real critical parts, and they're given the evocative, useful term names of complex 1, complex 2, complex 3, and then this one's called Fred. No, it's actually called complex 4. Depending on how you want to lay it out, we can also have a really different kind of structure that we'll get to in just a second that has a hole in it called complex 5. We'll see in a moment. This is in fact given the name ATP synthase. And as I say, I rebel at that term because it's not ATP synthase, it's ADP rephosphorylase. But hey, they don't ever listen to me. What do you want me to say? All right. So this is how it works. Complex 1 is where NADH backs up, beep, 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 like a dump truck, and dumps its electron. So NADH dumps an electron onto complex 1. And NAD is regenerated and given a little pat on the behind and told, go back out and pick up an electron. Like a good puppy dog, it runs back out and grabs another electron. Complex 1 hands its electron off to complex 3. Complex 3 hands its electron off to complex 4. Now, complex 4 is a really critical guy. It's handing its electron off to oxygen. So we're going to have O2 actually getting reduced. The electron is going to come here, and that generates a molecule of water. Now, a couple of critical points here. A, this is the only spot in all of your physiology that you need oxygen. You don't use oxygen anywhere else. It is only used as the final electron acceptor. In fact, so important, we're going to write that on the board. O2 is the final electron acceptor. All right, just so we can finish this up, complex 2 is where FADH2 dumps its electron. It doesn't have quite the high energy to deal with complex 1, so the electron is here. And then it passes it along as well. It just waits its turn. Okay? Yeah, it's a little complicated, but bear with me. Now, one of the cool things is each one of these complexes, complex 2 can't do this, but complex 1 and 3 and 4, they also had the ability, every time they grab an electron, they can take a hydrogen ion and shoot it out, pump it out. And they do this a lot, to the point that in a actively metabolizing mitochondria, in here we've got a pH of 8, and out here we have a pH of 7. Now you might say, oh, big deal, single pH unit. But remember, the pH scale is a logarithmic scale, so that means there's 10 times more hydrogen ions out here than there are here. It's a little bit like a dam holding back water. Now, the critical point to all this is, this outer membrane is permeable to protons. They can move back and forth. The inner membrane, though, is impermeable to hydrogen ions. It can't pass through. The only place that hydrogen ions can pass through is right here. So hydrogen ions zoom back through, and that is what generates the force to rephosphorylate ADP. And so this system is so evolutionarily conserved that in microbiology we draw the exact same diagram. So bacteria do this, you do this, elephants do this, plants do this, everybody does this. This is how ADP is rephosphorylated on this here planet. Cheers. Now, this is a really critical point because there's a lot of stuff that can happen here, a lot of ways to kill yourself. One way is stop breathing. That ain't going to go well. But there's actually a couple of other things you can do. Is there are molecules that will block the electron passage, and they do it pretty early in the process, such that you can't get the hydrogen ion being generated. Things like cyanide. That's where cyanide fits in, right around here. And so it will inhibit the flow of electrons. Taking cyanide is essentially the same as stopping breathing. And another really cool trick is a diet drug that is no longer actually much being prescribed. It is something called a nitrophenol. And it used to be prescribed back in the 40s, and it's a really cool trick. Let's see how it works. Or actually they're called DNPs, dinitrophenols. DNPs can fit into the membrane and punch little holes into the membrane such that the hydrogen ions can pass through. Now what that means is the hydrogen ions are not actually driving ATP synthesis. They're just allowing, the uncouplers are allowing the hydrogen ions to pass back through without generating ATP. Now, it does have some consequences because there's energy there. I mean, it was being used to make ATP. In this case, as the hydrogen ions just pass through, you actually begin to generate a little bit of heat. And people that take DNPs actually begin to run a slight fever. Now, as it turns out, when you have a fever, you're not generating it this way. You generate it primarily through muscle movement and things. But there are mitochondria that actually do have a naturally occurring uncoupler that are referred to as thermogenic mitochondria. They actually generate body heat. And little babies apparently have a lot of mitochondria that are that have some uncouplers in them. And so they can generate more heat because they're a little tiny and they can lose heat really fast. One of the things that it does, though, is that by putting these uncouplers in, you're not extracting all of the energy that your food has in the form of ATP. So it's essentially like taking your food and say a Pop-Tart's got 200 calories, you take a dinitrophenol tablet, now that Pop-Tart, while you're eating it, and it would normally have the ability to generate a lot of ATP, it's going to generate somewhat less. Now you have to be careful with this because if you take too much, you don't generate any ATP and you can't get enough energy from the food you're eating. But it is an amazing diet drug because you would have zero effects. You wouldn't even know that you're on a diet. It is simply you're not extracting energy from food as efficiently as possible in the form of ATP, and you generate it as heat. And so you run around with a little bit of a fever. But people end up screwing themselves up really badly. You can get a bad fever, that can kill you. You can also take too much and not get enough energy from the food you're eating to the point that you can't survive. Oops. So we don't prescribe those much anymore. Now, a couple of interesting things, though, can also happen, particularly if you decide to take a plastic bag. So go ahead and take your roommate, put a plastic bag over his head. No, don't really do this. If you block off oxygen, let's see what happens. So we got no O2. And this can happen surprisingly pretty easily. You can stop breathing, which is not recommended, but in rapidly metabolizing mitochondria, like in the big muscles of your legs when you're running, you actually can begin to run out of oxygen at a sufficient level to accommodate all the electrons coming through. So you're just not getting enough oxygen to those muscles. And this term is called going anaerobic. So what happens if you don't have oxygen present at the levels you need? Let's just look at this for a second. So with no oxygen, complex 4 doesn't have a place to dump its electron. Complex 3 now is ready to hand its electron off to complex 4. Complex 4 is still standing there going, I got my electron, I can't move, I don't have any room. Complex 3 goes, what the hell am I going to do? Well, complex 1 picks up an electron from NADH, passes it on to complex 3. Complex 3 says, I can't take it, I'm full. And complex 1 goes, well, what the hell am I supposed to do? NADH backs up and says, I got my electron for you. And complex 1 says, I'm sorry, I'm all full up. And NADH goes, uh-oh, what am I going to tell the boss? Because if I can't recycle back to NAD, I can't pick any more electrons up. This is going to be a serious showstopper, Hoss. And you better tell somebody to turn the oxygen back on. So what happens very quickly, within like 30 seconds, the NADH in your body can't be recycled back to NAD. And so all of the electron carriers end up being stuck in the reduced state. They can't cycle back to the oxidized state. And the first thing that stops is the Krebs cycle, because it requires a fair amount of NAD to be able to carry out its reactions. As that backs up, glycolysis is trying to stick pyruvate into the Krebs cycle, and it can't do it. We have an interesting little trick here that leads us to our next discussion about alcohol. And this is something that is known as anaerobic respiration, kind of also called fermentation. Let's look at this for a second. So if you recall, in glycolysis, we have a spot where we need an NAD, and we can make 2 ATP if we've got it, because glycolysis, as you recall, looks like this. We start out with glucose. We put in 2 ATP. They are not ADP. We split this guy into 2 3-carbon molecules. Each one, just note that, 3 carbons. Each one is now going through parallel reactions. The first thing that happens is oxidation. NAD picks up an electron, and it becomes NADH. This 3-carbon molecule now has the ability to generate 2 molecules of ATP, or to rephosphorylate them. This is, I can't remember if I mentioned this, this is where creatine can come in. And so creatine is potentially, theoretically, useful when you're carrying out high energy type of exercise where you're not going to be able to oxygenate quite as well as you should. Things like real rapid explosive movement, like weightlifting or sprinting, can lead into oxygen debt. And so that's why people would supplement with creatine, because it helps with this step where you're going to take 2 ADP and generate 2 ATP. And you're left with pyruvic acid, or as it's also known as pyruvate. Let's just fill this side out as well, just for completeness. So we've got NAD picking up an electron to become NADH. We've got 2 ADP coming in and generating 2 ATP, and you got pyruvate. Now, so we have 2 of these. In the absence of oxygen, this NAD isn't going to be present. It's all in the form of NADH. So you stop. But we have this evolutionary trick that's like this. And this is a good way of sort of defining it. If we define standard respiration as aerobic, we can say that O2 is the final electron acceptor. But we've got a slight sort of side branch we can do. Doesn't work very well, but we can do it. And we can use pyruvate as the final electron acceptor. And this is known as either anaerobic respiration, also known as fermentation. And let's see what happens here. What we're going to do is take pyruvate, dump the electron from NADH, so we're going to reduce pyruvate. NADH dumps its electron, becomes NAD that cycles back up there, and we have now reduced pyruvate. And the form that it takes depends on what kind of organism you are. In animals, we generate the 3-carbon lactic acid. This is the acid that begins to form in your muscle cells after intense exercise. And this is why your muscles get sore. Now, muscle soreness is actually a sign that you're not getting enough oxygen to your muscle cells. Now, don't be alarmed. One thing you can do is, as you exercise more, you can increase your oxygen carrying capacity, and you can provide oxygen to these cells better. Second, if you could do your exercise with a little tube under your nose running pure oxygen, man, you'd be golden. You would never get sore. You could really run a lot better, work a lot better. Presumably that's against NCAA rules, but if you could somehow sneak an oxygen tank into your kit, like while you're running, oh honey, you'd be feeling a lot better and you could go a lot harder. Well, that's lactic acid. Another way that is carried out is taking pyruvate, and pyruvate then can lose a molecule of carbon as carbon dioxide, and we're now left with a 2-carbon molecule that gets reduced. NADH dumps its electrons here, so let's put the NADH and its electron. And you now make the 2-carbon ethyl alcohol, or ethanol. And this is the principle behind yeast fermentation. So we can't do this reaction, but yeast can. And so when you put yeast into a solution with sugar, like grape juice, and let it sit for a while, fairly quickly it begins to deplete the oxygen that's in the liquid, in the juice, and it then goes anaerobic. And when it does, it begins to produce ethanol. Now, a couple of interesting points here is that this is not a great way to operate because, first, you're generating a rather nasty toxic byproduct. With oxygen, you generate water. Water is lovely. Nobody's going to bother with having water as a toxic byproduct. That's a nice metabolic byproduct. But lactic acid, that's bad news. If you do it too much, you can actually begin to lower your blood pH and kill yourself that way. Lactic acidosis, not good. And it's also not a very efficient way. With oxygen, we get 36 ATP. Without oxygen, you make 2 ATP net gain. Whoa! 36 versus 2, I'll take 36, thank you. What this means is if you're a multicellular organism, you can't operate without oxygen. Single-celled organisms, bacteria, yeast, they can operate without oxygen, mainly because they're just a dot. They're standing around. They don't get a lot of ATP. You know, it's like, hey, if the oxygen's on, they're getting lots of ATP per glucose, so they don't have to eat very much, and they can start doing their business. But with no oxygen, they just have to stand around and accumulate ATP over, a period of multiple hours as opposed to doing it in 15, 20 minutes. So just another way of thinking about this is if we've got cells that are like yeast cells that are dividing, and say they need 50 ATP per minute to do what it is they do. That means that with 36 ATP per glucose, they just need to eat 2 glucose molecules per minute. And they've got more than enough to do what they need to do. But in the absence of oxygen, they now have to start consuming a huge amount of glucose to be able to do what they do. So rather than having 1 glucose molecule per minute, or wait, what we say, 2 glucose molecules per minute, they now are having to consume something like 25 glucose molecules per minute. So their glucose uptake can be dramatically increased. But when you turn the O2 on, the glucose uptake drops dramatically. The other side of this is that the yeast in your grape juice will eat up the glucose very quickly because they're going anaerobic. And they're going to be converting it to ethanol. So, the quiz I'm building in Moodle again. I'm not sure exactly when it'll drop. Maybe tomorrow, maybe on Monday. Anyway, I'll give you next week to take it, so don't worry about that. This is the nuts and bolts of what we're going to be covering for this next quiz bit. And we'll see where we go from here. So the next topic we're going to get into is, starting with ethanol, start thinking about fermentation and the creation of beer and wine, and then look into distillation to make more concentrated alcohol products, and then finally, from there, get into why beer makes you pee. Won't that be exciting? I'm sure. Well, that's what I have to say for now. So hopefully you watch this, go through it, take some notes. I'm going to give you some handout stuff too, supplemental stuff, and you'll be ready to do the very easy quiz. That's all I have to say. I'm out. Peace.